All right, hi everybody, this is Lilith and I'm one of your IAs for BIPN 160. This video will be a little bit shorter because I will cover the first part of lecture 10 and then a little later tonight I will cover the second part of lecture 10. Okay, so in this video I will go over the first half of the lecture that Dr. Root gave on lecture 10, and he basically talked about the mesolimbic dopamine pathway. And for you to understand the mesolimbic dopamine pathway, let's go over the key parts of this pathway and what each one of them does. So I like to start by looking at the ventral tegmental area, or VTA for short. So the VTA is located right here, and it projects dopaminergic neurons all the way to the nucleus accumbens, which is right here. Okay, then the nucleus accumbens will send information to, or project neurons to the ventral pallidum, right here. And the nucleus accumbens will also project back to the ventral tegmental area. So you can think of this circuitry, or this mesolimbic dopamine pathway, as a circle going from VTA to nucleus accumbens, then back from the nucleus accumbens to VP, and back to ventral tegmental area. We will look at a schematic that shows this a little better in the next 2 minutes or so. Okay, and the important thing here is that the ventral tegmental area projects dopaminergic neurons, and these are neurons that release dopamine in the nucleus accumbens. Okay, and why are we talking about the mesolimbic dopamine pathway? Well, this pathway is involving structures that are part of the limbic system. I think the nucleus accumbens isn't technically part of the limbic system. However, other parts of pathways that we will discuss are. Okay, and the limbic system is important in emotion, liking or disliking things, feelings, and so on. And all you need to know about the limbic system is that it contains a lot of parts to it. For example, amygdala, hippocampus, cingulate gyrus, and so many others. But for now, you can only keep amygdala and hippocampus in your mind. Okay. Now, the rest of this lecture, we talked about specifically the VTA, then the nucleus accumbens, then the VP. So let's start talking about the VTA. So as I stated earlier, the VTA becomes activated, and when it does, it releases dopamine from its dopaminergic neurons, and it stimulates the nucleus accumbens. There are two types of activities the VTA is involved in, and those two are reward prediction and learned reward association. And so let's go over each one of these. In this picture right here, or this experiment right here, we talked about how when there is an animal model, for example, let's say a mouse, I think it was a mouse in this experiment, if the mouse is given a droplet of sweet juice, something that is a reward to this animal, then we see the firing of the VTA dopaminergic neurons right here, right after the juice is given. So after the juice is given, they feel the reward, and that activates the dopaminergic neurons in the VTA. However, if you start to pair this reward of sweet juice with a light signal, then the animal begins to realize this and begins to understand that when it is given the light signal, moments later, it will also receive a reward. So, slowly throughout time, the mouse begins to have its VTA neurons activated right after the light signal is given. And this basically notifies the fact that this animal now expects to get this juice. So because the dopaminergic neurons in the VTA are activated right after the light signal is given, tells us that these neurons are involved in reward prediction and not so much the reward itself. Because you see how over time, yes, these VTA neurons were being activated for the reward itself, but after the mouse understood that it can predict when the reward is going to happen, this activation of the VTA over here stopped happening because it would happen much earlier, right after the signal was given. Okay, so this indicates reward prediction. And this was further confirmed by the fact that if the researchers kept on giving the light signal to the mouse, however, they stopped giving the mouse the actual reward, then although the VTA would activate over here after the light, when the mouse waited these moments to get the juice, and it never did, these neurons actually became inhibited, right? They became hyperpolarized. So instead of, so the opposite of activation took place when they realized that over and over they are not given the juice. Okay? So some research data showing that the dopaminergic neurons in the VTA are involved in reward prediction. Okay. The research study that showed how these neurons are involved in learned reward association was this study. And in this study, I believe all that you need to understand is that they trained the model animal for a learned task. So they taught the animal to do something, right? To learn to do a task. However, when they lesioned the VTA, when they lesioned the nucleus accumbens, meaning there was nowhere where the dopamine could arrive to from the VTA, then they saw a big decline in the mouse's ability of doing the task that they taught it to do. Okay, so again, if you're following the theme of the class with if you lesion an area or if you inhibit an area of the brain and the function declines, it means that that area of the brain, in this case the nucleus accumbens, which receives dopaminergic neurons from the VTA, this area is needed for the mouse to be able to do a learned task. Okay. Before I move on, this is the diagram that gives you a better understanding of this circuitry or this cyclical activation and inhibition in the mesolimbic dopamine pathway. So to break this down, this here is the VTA, and the VTA projects to the nucleus accumbens, and then the nucleus accumbens projects to both the VP and back to the VTA. Okay, there is also input into the nucleus accumbens from the prefrontal cortex. And we didn't talk much about it, and we don't in this lecture, but that's something that Dr. Root mentioned over here, so that's maybe something you want to at least be exposed to. Okay, and then another thing you want to remember about the inputs is that the VTA has dopaminergic neurons, meaning they release dopamine into the nucleus accumbens. Now, in the nucleus accumbens, there are many different types of neurons. There are interneurons, there are D1 neurons, and D2 neurons. The D1 neurons are responsible for positive stimuli. And I believe they stimulate the next area that they're projected to. The D2 neurons, over here, respond to aversive stimuli, and they can inhibit the next area they project to. Okay, so there is a large theme of valence in this lecture, and valence means liking or disliking different stimuli. So you will notice that in the nucleus accumbens, we have two types of neurons, and one of them responds to positive or appetitive stimuli, meaning something that the mouse likes and maybe something that smells good or tastes good. And then the other types of neurons, in this case the D2 neurons, are responding to aversive stimuli, so something that scares them or something that disgusts them. Okay, and then the other important thing from this diagram would be for you to memorize where D1 and D2 project to. So remember that D1 projects to both D1 projects to both the VP and VTA, and then D2 projects to the VP, and then the VP projects to the VTA afterwards. Okay, so we have the direct pathway with D1 and the indirect pathway with D2. When we look at the nucleus accumbens by itself, we can see that it is organized in a sense. And by organized in quotes, I mean the anterior portion of nucleus accumbens responds to positive or appetitive stimuli, and then the posterior side of the nucleus accumbens responds to aversive stimuli. And the research study that was done here, I don't think you need to know the details, but if I remember correctly, they gave the mouse a whiff of something that is aversive to them, and then they also gave droplets of sugar water or sugar-infused water, and that is obviously a positive stimulus for them. And so depending on which type of stimulus they gave to the mice, different areas of the nucleus accumbens were stimulated. Okay. Now this study over here showed that different neurons respond to different stimuli in the nucleus accumbens, and that's kind of this idea of having the D1 and D2 neurons. Remember how I just said that some of them can activate and some of them can inhibit the next brain areas? So for example, I think D1 can activate VTA and VP, but D2, when it activates VP, VP can inhibit VTA. I don't know if I said that right. But maybe scratch that. All you need to know, I think the basic or the essential point that you can take away from this is that in the nucleus accumbens itself, there are different neurons and they respond to different stimuli. And by responding to different stimuli, they can either activate or inhibit And by being able to do both, you see how these lighter parts of this diagram, that means activation, and the darker parts mean inhibition. And by activating some parts and inhibiting other parts, you create kind of like a unique map, which shows a unique understanding of the stimuli that you're getting. I feel like I butchered that, but please email me if that didn't make sense. But the basic point here, again, would be that different stimuli, different neurons in the nucleus accumbens pick up different stimuli. And there are neurons that activate, and there are neurons that inhibit. The nucleus accumbens is also an important site of cocaine effects. So the way cocaine works on the dopaminergic neurons coming from the VTA is cocaine blocks the reuptake machinery of dopamine. So this neuron, after it releases a lot of dopamine into the synaptic cleft, if there's cocaine present, it can no longer take this dopamine and retake it back into the neuron. So imagine if there's cocaine, then all of these dopamine molecules are just going to stay in the synaptic cleft and just overactivate your nucleus accumbens. And in this study over here, they had a catheter that would just give the mouse cocaine whenever it pulled the lever down. And here it shows how the mouse can self-regulate how much cocaine it wants and how stimulated it wants to be. So if they reduced the amount or the concentration of cocaine that was given through the catheter, then the mouse would push the lever more. And if they increased the concentration of the cocaine, then the mouse would push the lever even less because it was regulating how much cocaine it wanted to get. And then in this last portion, they gave the mouse a molecule that blocks the dopamine receptors in the nucleus accumbens. And so imagine if those dopamine receptors are blocked, then this dopamine will not have an available receptor to attach to. So it doesn't matter how many times the mouse pushes this lever. Yes, there is dopamine being produced, right? And this cocaine is blocking these reuptake machineries. So there's a lot of dopamine in the synaptic cleft. However, the dopamine receptors are blocked. So the infusion of cocaine is not really doing anything to the mouse because the dopamine receptors are blocked anyways. In this diagram, it shows how different drugs or different stimulants are affecting which part of the mesolimbic dopamine pathway. So for example, cocaine, as we just learned, blocks dopamine reuptake, and it stimulates the dopaminergic neurons by stimulating the ventral tegmental area, nucleus accumbens, and the prefrontal cortex. Okay, and so do amphetamines and MDMA. However, nicotine and all these other ones work differently. And you definitely want to look at the definitions here and how they work, because this was a type of a question that you were asked in lecture. So I'm guessing it could be a question on the exam. Okay. Then we talked about the study done on humans, where the scientists wanted to see if getting rid of the nucleus accumbens will actually help people who were addicted to drugs. And I guess another question that you can ask by this is nucleus accumbens required for developing drug addiction? And again, if you're trying to find out if an area of a brain is required for a certain function, then you want to lesion or inhibit that area of the brain. And if the function is reduced or there's a decline in that function, then you know that that area was required. So what the scientists did in human brains, which was very risky and illegal, is that they lesioned the nucleus accumbens of these people. And indeed, it showed that the people who had lesioned nucleus accumbens had much more abstinence levels. So they had less relapse and becoming more free of drug addiction. Okay. If you remember back when we talked about this diagram, then you remember that the nucleus accumbens also projects to the ventral pallidum, the VP for short. So let's go back over here and let's talk about the type of neurons that the VP contains. So this is just another diagram of this circuitry. So over here we have the ventral tegmental area. Okay, it projects to the nucleus accumbens. Then the nucleus accumbens projects to the ventral pallidum and also to the ventral tegmental area, which is not shown here, but it also completes the circle back to the VTA. And then the VP also projects to the VTA. And the important types of neurons here are the two types of neurons that again respond to either positive or negative stimuli. So it is found that the neurons in the VP that are glutamatergic, meaning they release glutamate, they respond to either punishment itself or punishment cues, meaning that there's a cue that signals that they are going to have punishment soon. And then the GABAergic neurons, which release GABA, respond to reward or reward cues. The key VP outputs that Dr. Root put over here, you definitely want to be exposed to this or to remember it. The VTA is easy to remember because, again, this is in the cycle. The VTA projects to nucleus accumbens, and nucleus accumbens projects back to the VTA. So that's one of the outputs of, the ventral pallidum, the VP, projects to the VTA. So VTA is one of the outputs. And then I guess it also outputs to the thalamus, but we didn't really talk about it. And then the lateral habenula, we didn't talk about. So I doubt he'll ask you, but it's there if you need it. But I think so far for the exams, Dr. Root has mainly asked questions that he has talked about. Maybe not all the time, but mostly. So I don't expect him to ask anything that he merely mentioned once. Okay? But then to finish off this video, I want to talk about the two experiments that we talked about. And these two experiments are regarding the two types of neurons in the VP. So as we just said, there are two types of neurons in the VP, glutamatergic and GABAergic. So in this experiment right here, they took VGAT, I think it was just a protein that is present in the VP. So they attached Cre to it, and then Cre was able to induce the expression of channel rhodopsin, and channel rhodopsin was expressed in the GABAergic neurons of the VP only. Okay, so that is the first step in this experiment. Now that we have channel rhodopsin in the GABAergic neurons, then we can stimulate the GABAergic neurons by shining a light on the channel rhodopsin. So that's exactly what they did. They made different areas for the mice to be able to poke their head in, and in the different holes, they would shine a different frequency of light. And the higher the frequency of light, the more channel rhodopsin you would activate in the brain of the mouse. So they would let the mouse free, and they realized that most of the time, by an overwhelmingly significant number, the mouse would prefer to stick its head in the hole that would have the highest frequency of light. And why is that? This tells us that the activation of the GABAergic neurons is something that the mouse likes, or else it wouldn't poke its head into that hole to get that activation. So this further confirms that GABAergic neurons are giving the mouse the sense of reward and a feeling of liking what that feels like. Okay, so this is what that first part of the experiment says. And then another part that Dr. Root mentioned was this part, and this is just localization for the mouse. So if this right side of the box is an area where they shined a high frequency of light, activating the channel rhodopsin in the brain of the mouse, which will in turn activate the GABAergic neurons, then you see how the mouse would spend a lot of its time in this area of the box because it liked the feeling it got when the GABAergic neurons were activated in its brain. Okay, so again, I just saw this, it's activation of GABAergic neurons causes place preference / liking. So they like to be in this area where they're getting a lot of the GABAergic neuron activation. Over here on the right, this experiment talked about the glutamatergic neurons in the VP. And here they used another gene of possibly a protein, or yeah, it's probably a protein. And they attached Cre to it. And then as you remember, when Cre is expressed, then it can remove a stop codon by using the LOXP site. And if you remove a stop codon, then you can express channel rhodopsin. In this case, they expressed it in glutamatergic neurons of the VP. And again, they shined light in this right side of the box. And when they shined a high frequency of light, they activated the channel rhodopsin in the brain of the mouse. And this activation caused activation of the glutamatergic neurons. However, they saw that the mouse did not like to stay here, and it completely avoided the side of the box that was being activated. And instead, the mouse was hanging out over on this side. This told the researchers that the glutamatergic neurons are actually involved with a feeling that the mouse doesn't like, right, with aversion or possibly punishment. And so that further confirms the fact that the glutamatergic neurons in the VP are responding to a negative stimulus, punishment, or aversion. Okay. I think I will stop here for this video, and I will go over part 2 of lecture 10 in the next video I post. Okay, I will put the PDFs of each video separately in my IA page so that you can find them easily in a more corresponding way. If you have questions, my name is Lilith. Please email me. I've been having a little bit of a busier week than usual, but I will try to get back to you as soon as I can. Okay.